Hello everyone. Today we will understand about basics of flow cytometry. So what is flow cytometry? It is a technology which is used for cell counting and cell sorting. Going in depth of the definition, it is a laser or a impedance based biophysical technology used in cell counting, cell sorting, biomarker detection by suspending these cells in a stream of fluid and then passing them through an electronic detection apparatus. To understand this definition, we should understand what are the principles of cytometry. So what are the principles? How does it work? This is very important to understand about flow cytometer. So firstly, there is a cell sample. Okay, now this cell sample can be blood, it can be bone marrow sample, it can be any aspirate, it can be also any solid tissue which can be converted into single cell preparation for the technique to process. So this is the sample which goes inside this, and now this is a nozzle here. This nozzle will convert the sample into single cell column. This is single cell column. Each cell is examined. Now the cell falls at a specific speed, and for that each cell can be examined. This was a flow system. Second is the optical system. The laser light, it is emitted and it falls on the cell. Now when it falls on the cell, there is a production of a scatter. There is scatter, we will understand it later. The scatter, this light goes over here, this light goes over here in various on the mirrors, and then it is picked upon by detectors. All these are detectors. They detect. What are they? PMT means stands for photomultiplier tubes. They will pick up the scatter and then they will convert it into a current and a volt along with voltage, and then it is sent to a digital converter. And when this there is a series of softwares, and then the PC, there's a graphical representation of the various quantity of cells present in sample. So this is all about the flow cytometer. There is a flow system, there is an optical system, there is an electronic system, and then there is a computer system. Okay, this was about the flow cytometer. This we have understood. There are 4 systems. Now common specimens which we will use for flow cytometry are: there will be peripheral blood, bone marrow, body fluids, cerebrospinal fluid, urine, lymph node aspiration, and even the solid tissue if we can convert it into single cell preparation, which is done with the help of various enzymes like collagenase. Then what is the concept of scattering? We understood that when the single cell preparation is falling down, there is laser light falling on it, there will be scattering. What is it? Now what is scattering is that this is a cell. Okay, we are seeing this is a cell. Okay, this is its nucleus. Okay, so this is the light falling. It falls on the 180 degrees and then it scatters. Okay, this is the scatter. Now this scatter, known as forward scatter, will tell us about the size of the cell. We will understand about the size of the cell by this. Okay, you can see this is telling us about the size of the cell. And then there is a side scatter. The light falls on a specific organelle, specific granule, and the side scatter takes place. Now the flow cytometer detects both the side scatter and the forward scatter. Forward scatter will tell us about the size of the cell, and side scatter will tell us about the internal complexity of the cell, on the basis of which a graph is produced. This is a graph which we are seeing here, and this is a side scatter. Here we will interpret the forward scatter, and various cell populations are interpreted. Now to understand this, now this population of cell falls over here and over here in this margin. So it has lower forward scatter and it has lower side scatter, means cell is small and has a low internal complexity like that of a lymphocyte. This cell here It is larger in size but has no internal complexity. It also has internal complexity almost same. So it can be like, it can be monocyte. So this is how we will understand about the scattering. Okay, so there is a forward scatter. It collects the light at 180-degree, and it tells about the cell size. In this, how we will differentiate? We will differentiate into normal cells, monocytes, neoplastic cells on the basis of cell size. And the size scatter will tell us about the cytoplasmic granularity and nuclear configuration. Example is like that of neutrophil. The site scatter of neutrophil is higher because it has a higher quantity of granules present. This is again same picture showing the difference between the side scatter and the uh, forward scatter. Okay, now the parameters analyzed by flow cytometry, we can understand from here that it uses the physical properties of the cell, the size, the cytoplasmic granularity, and also the cell antigens. To understand about the cell antigens or markers, specific antibodies are needed. These antibodies can differentiate the cell type, the cell lineage, and the cell maturation stage. This is known as immunophenotyping, and for this we require antibodies and also the compound known as fluorochromes. Now for antibody, we will require highly specific monoclonal antibodies, and these antibodies nowadays they are based on cluster of differentiation, the CD markers. The CD markers they are used, and they are used for identification and a distinction of the cell surface antigens according to that. And what are the fluorochromes which are used? Fluorochromes are mostly used along with the antibodies. What happens is, this is a molecule of fluorochrome. Fluorochrome is excited by a light source and it emits a fluorescent wavelength. For that we will understand it like that. What happens is, the when we have this cell and there is a fluorochrome attached, the fluorochrome the wavelength by which it is excited, it gets excited and then it emits its wavelength. Okay, this wavelength we check for this wavelength, the emission wavelength. So the antibodies they get conjugated to the fluorescent dyes. Okay, we will this is the normal sample. In this the antibodies get conjugated with the fluorescent dyes, and now when this is passed through a light source, there is emission of the wavelength, and this emission of the wavelength we will check for. When the labeled cell they are passed through the light source, the fluorescent molecules are excited upon high energy state, and upon when they return to their resting states, they emit a light energy of higher wavelength. This is known as emission wavelength. Okay, this was known as the excitation wavelength, which was this one. This is excitation wavelength. Then this wavelength, emission wavelength, we check, and we according to that we will interpret it. Okay, by this we will understand about the immunophenotype of the cell. Example of the fluorochromes which are used are many. There are many fluorochromes. Just examples: phycoerythrin, Texas red, fluorescent isothiocyanate. We can see the excitation wavelength may be the same in various, but the emission wavelength is different. And here from this we can understand the various cells present. So this was about the flow cytometer. We should revise the basics of the flow cytometer. There is a sample, there is a flow system, then there is a single cell column of the cells which upon hitting with a laser light they emit it, and the photomultiplier tubes they get the signal, and then the software tells us about various types of cells present, whether they are immunophenotypically labeled or not. Okay, this was about the flow cytometer. We will discuss about applications of flow cytometry in the next video. Do like, share and subscribe to my channel. Thank you very much for watching it.